Bioinformatics. Okay, so this is going to be a follow-along video for accessing databases. This was our third lab, and this lab will cover accessing information and pulling it down into your server from both NCBI as well as the short read archive. So first things first, I'm already logged into my server. Again, follow videos that I previously made available to you if you forgot how to do that. First thing we're going to do is we're going to make a directory for us to work in. I never like to just work in my home directory. So I'm going to type ls, and you can see, I have a little bit of organization here. I've got the lab that I made the last video for you on, which was lab 2 on the FASTA, FASTQ format. So let's kind of follow that theme, and we'll make a lab directory called lab 3, and we'll cd into it. Remember, we're going to be using tab completion and hitting enter. So I'm not going to go through and read all the information that you were able to read during your kind of first attempt at this lab, but I'm going to go through most of the code and talk about it. So one of the first things I want to show you is how, if there's a publicly available genome and it's been made available just to download, the easiest way to download things is with wget or curl, which we're already experienced with. So we could curl -big O, not -0, this URL to this FTP, and download this Homo sapien genome here. This is for chromosome 38. And it's small. It's also zipped as well, .gz. Recall from our intro to the shell for genomics, we could unzip that file there if we wanted access to it right away. We're not going to be doing that for this lab, but just these things all kind of tie together. You could even head that file if you wanted to and see that it's in a format that we haven't been exposed to yet, but it's very much a good file that we downloaded. So moving on down, there's a variety of ways to access the NCBI data sets. We're going to be first looking at how to use accession numbers related to projects or individual sequences or genes, and there's several different ways to do that. If you wanted to do this kind of on your local computer without doing any code whatsoever, the Entrez has kind of this nice kind of URL function where you can go to this service.nih.gov and really kind of fill in these blanks where I'm saying value, value 1, value 2, value 3, and you fill in related to different parameters. And you just type that into the URL and you'll get what you want. So as an example, here I've put in an example where we've got this long URL, and our first parameter is the ID, and it's set equal to this value 1. This is the sequence ID. The database type, DB, this is set equal to the nucleotide core, and then the actual type of file that we want, which is FASTA. Okay, so you could actually copy and paste this line into your browser and access it that way. But again, we're working within our servers, so why don't we do something different and actually curl it down and check it out in our servers. So here, what I'm doing is I'm curling it and then I'm piping it, recall from the shell for genomics, and I'm just going to look at the first 10 lines of it. And so what do we have here? Well, this is in FASTA format. So we've got a 2-line format where the first line is some kind of indicator. Now this is an assembled genome. It's still okay. It can still be in a FASTA format. Even if it's super long, it could be a gigabase. And it could still be just 2 lines. It could be that first line that says what it is, and then the second line could be an entire chromosome or an entire genome. And so here we've got just, it's wrapped around, but it's actually really the equivalent of 1 line all the way down. And that's the whole sequence for the Ebola virus, for this sequenced accession of it. Again, there's multiple sequence genomes, many sequence genomes of many viruses. Okay, so that was kind of a quick and easy way to download genomes and other NCBI accessions using just kind of plug and play with a URL and downloading it either through your browser or through kind of curl or wget. But that's not really what we're going to be doing most of the time. That's really not very scalable. Instead, we're going to be using kind of packages or applications that are pre-built to kind of ripping things down from these sources. So like many things, we're going to be installing using Conda. We're going to install the Entrez direct utility. So this is conda install -c for the Bioconda repository, and we're going to install Intrez Direct. And it's going to think for a minute, and while it's installing, we'll go ahead and look at what we're going to be doing next. I always kind of look ahead during these things. We're going to be using it to kind of use those same three pieces of information that we looked at with the URL. We are going to provide information on what database we want information from, the format we want, not FASTA this time, and then the ID, in this case, a slightly different ID. And then we are going to redirect the output because it's going to print the output directly to our terminal, and I'll show you that. But we actually want to redirect it into a file so we can use it later. Sometimes it's good to have it come to the terminal, though. So let's go ahead and test out this eFetch. eFetch tab complete whenever possible. And you could copy and paste this, or you could type it. You're not going to be able to tab complete these kinds of things for the most part. So this is where sometimes copy and pasting can be helpful, especially things like an ID so you don't mess it up, but I'm just going to risk it. So AF086833, and I'm not going to redirect it to a file yet. Let's go ahead and hit enter and look at what it looks like. Okay, so if we scroll up, this is our genome here. And this is in a GenBank format. And so we can talk more about that later, but in addition to the sequences, we also get extra information about what the translation is, inferred annotations, and all sorts of good stuff. These are a little bit harder to work with because they're more complicated file formats, but they have a lot of information in them. Okay, so let's redirect that. And we wanted to redirect it to the accession ID number 86.gb. 86833.gb. We'll go ahead and hit enter. Okay, now if we ls, we've got that file in there, so we can use it whenever we want. Now, you can manipulate the values for each of the different parameters when you use eFetch to get different things. Let's say that you really hate GenBank format and you don't need the annotations. Well, let's try and get a FASTA. It is as simple as going here and saying FASTA. Now, how did I know it was FASTA and not FA? Trial and error, or you could Google the manual for eFetch, or you could look for eFetch, see if it has a help function, or you can just kind of remember. You know, there's only several formats that we're going to be using, and it's just kind of one of those things you pick up as you go, or you take notes, or you just Google it every time you have to do it every once in a great while. All of those are acceptable options. Okay, so let's head what we just downloaded. So af.fa, and yep, that looks like a FASTA format to me, right? Okay. So there's other things that we can do. I just showed you kind of the three primary parameters. We can also look at subsets of sequences, both from starting points and stopping points. So if we only wanted to look at the first 5 base pairs, we could execute that right there by adding seek start and seek stop at the base pair. So here we have just the first 5. We could also add what strand we wanted, whether we wanted strand 1 or strand 2 from the reference. And so we could sequence we could execute that. And again, strand 1 is what we get just by default. But what happens if we put strand 2? Well, it looks like we're getting a complement. And so I kind of have a note in here to kind of look closely and make sure you always think about what you're doing, because there's 2 ways to complement things. You could complement them without reversing it, so that you would have a C and a G, then a G and a C, then a G and a C, then an A and a T, and then a C and a G. And it almost looks like that. But this AA kind of gives it away that really, and I have it printed down here for you, this is actually a reverse complement. So it's always important to know exactly what you're asking for. Are you asking for complementary bases, or are you asking for reverse complemented bases? Okay, so let's say you didn't know the exact sequence ID, but you knew there was a paper you were interested in. You looked through, and like a good paper should do in genomics, they provided their project accession number. Not sequence accession number, project accession number. Typically starts with a PRJ, but that can vary. let's say that we found this PRJ in some manuscript. We can try and find all of the protein sequences, all the nucleotide sequences, all the associated with that project. And so there's ways that we can query for those in a nice, helpful way. And so here's an example of using eSearch, looking within the nucleotide database and querying for a project ID. And so this looks kind of funky. This is actually an XML format. Don't get too hung up on it. It's actually kind of, self-explanatory. This line, this is DB for database, and then /DB is saying close kind of that window, basically. So this is a nucleotide within the database window. Then you could look at the number of things that it found here. The count is 249. And we could do the same for protein, right? So we could either copy and paste that, or we could hit up and just type protein here. Let's see how many. 2,240 proteins, okay? So we can use this little piece of kind of XML information about the project to get more out of it, because as it stands, this isn't really what we want, right? Maybe we want to know what those sequences are. So we can actually pipe the output of this, the XML, which is nice because it's in that weird format. It's readily accessible for eFetch to use. So we could pipe that right to our next command, which is eFetch. And let's say I kind of like to look at things in FASTA. So let's look at it in FASTA. And, well, we could redirect it to genome.fa as a new file that's going to contain all the outputs. But I always, when I'm building these kind of long pipes, I like to look at the output of each one individually before I have it go somewhere permanent. So let's hit enter and see what we get. And this might take a minute. Okay, so we've got tons of stuff. Looks like we've got some protein coming. One of those 2,240 proteins from this project. And this would probably print to the screen for a long, long time if I let it. So I'm going to hit Control-C to stop it. We know it works. That didn't go anywhere, so let's send it somewhere now. So we're going to redirect it to genomes.fa. Okay, it's going to pause for a second while it kind of slowly fills up that file with the output. I'm going to give it a minute, and I'll be right back. Okay, I'm back. And you can, for yourself, go ahead and look at genomes.fa and make sure that it's not an empty file. Sometimes you end up with empty files. If you were to do, I don't want to mess it up too bad, but let's just say you had like a typo where you said FATSA instead of FASTA. And I'll give it a different name, genomes1, and you hit enter. Okay, it's going to think for a while, and I'll let it go for a second. With our kind of bad example, it kind of kept going, and eventually I stopped it. And if we go ahead and head it, we have that file if you ls and look. But if we head it, there's nothing in it. It's an empty file. So sometimes whenever you have problems, always make sure that what you're doing is actually what you think you're doing. Make sure that when you're putting together complex strings of pipes, that you're not having a broken pipe somewhere in the middle that's making all the downstream files that you're going to be using unusable. So that's kind of a troubleshooting 101. Okay, so that's how we get data that's a little bit more processed. But what if you want the raw data fresh off a sequencer and you want to rerun analyses that another paper did from scratch, or maybe a new analysis has come up and you want to reanalyze data that were generated? Most papers are making their raw data available. In fact, many journals require that they make their raw sequence data available in as unedited a fashion as possible, often before it's ever been trimmed or anything like that. So, how do you get those data? Well, they usually are housed within the short read archive. And so the short read archive we can access very easily with yet another tool. So we're going to conda install from the bioconda archive SRA tools. It makes sense. It's tools to work with things from the SRA. In this case, if you've been following along, you probably already have the SRA tools installed as a part of the Entrez Direct utilities. But if you didn't, or if something weird happened, that line there will install the SRA tools by themselves. Okay, so the main thing we're going to be using from SRA tools is the function fastq dump. This is just dumping all of the fastq reads from any given accession number into your computer. So let's say that we have this short read archive accession and we want it. It is as simple as typing fastq dump srr 155 3607. I'm going to hit enter. Did I type that wrong? Yes, I did. I put 3 5s. There we go. Now, these files can be massive. These can be big sequencing files. And I tend to, give you guys smaller ones, but just know that this could take a long time. And even the smaller ones can take 5 minutes or so. So I'll be back when this one's done downloading. Okay, so I'm back and my file has finished downloading, and we can type ls just to see that for ourselves. We can head that file, again using tab completion to make it easy, and see that it is indeed in that 4-line FASTQ format. So we've got that. Now, something I didn't tell you about this file is that this file is actually from paired end sequence data. And again, we haven't learned too much about the difference between single end and paired end, but know that paired end are typically in two related files, typically designated with an _1 and _2, because you can think of them as, for any given read in there, as having a mate or a pair in the other file that is connected by some length of unknown base pairs. Often the length itself is known, but the base pairs between are unknown. And so, in short, if we're thinking about paired end reads, typically there's a DNA fragment, and you have forward reads coming from one side during sequencing and reverse reads coming from another. Sequencers, especially the short read sequencers, only sequence a certain number of bases reliably, so maybe it's 200, but if this fragment is 1,000, you could sequence 200 from the front and 200 from the back, and then have about 600 unknown bases in the middle, but you know that it's a fixed distance between them. So that can be important information when trying to reassemble sequences. For you right now, that's not important, but I just like to kind of throw that out there so you have a little more understanding as we go forward. But you're like, wait a second, they only gave me one file. So FASTQ dump by default will actually stick them all together in one file. The way that you make sure that it gives you all of them is if we go all the way up to our original line and we do -- split - files, it will split them for us. Notice that was a lot faster. Same amount of data. This is the good thing and a bad thing about the SRA tools. It will remember and save a backup of the SRA files that you download. And so if you go to re-download, either for splitting purposes or for subsetting, as we'll see here in a little bit, it doesn't have to go back to the internet and pull it all down again. It can just use what it already has. The problem with that is every time you download a new accession, you actually download it twice, and they can be quite large files. So let's take one detour real quick, because I want to show you in the home directory where the backups are housed, because it's good to delete them when you start to get low on space or when you're not going to use files anymore. So deleting them, both the FASTQs that you work with in the folder as well as where they're backed up at. So if we go to our home directory and type ls, we'll see there's this NCBI folder here. NCBI houses the SRA, so that makes sense. So let's cd into NCBI and ls. And again, we can do this in a quicker way, but we'll kind of take our time. Cd into this folder public and ls, and there's a folder SRA. Now if we type ls, we can see there's an SRA file associated with that accession number that we just downloaded. So what I would do is I would go into this folder and I would actually rm things star.sra, just to remove all these SRA backups. Okay, but we don't need to do that right now. We're working on these things, so we'll go back to lab 3 and continue on the path. So let's ls, and we can see that after we did our split file, we've got the _1, we've got the _2 for the paired end files. What if, for some reason, you only wanted a certain number of reads? Well, you can also, let's scroll up here real quick, get back to our command, not only split files, but you can use the -bigX to designate how many reads you actually want. So if I wanted 10, let's say 100,000 reads. So if I wanted 100,000 reads, I could do that and hit enter. And now I've got, actually I think that was 10,000, but whatever. So that's 10,000 reads right there. Something to keep in mind when you do this is it's not a random sampling from the actual sequence set. It's from the top down. So if these reads are in the order they came off the sequencer or something like that, there could be a lot of bias associated with that, or quality that is not representative of the rest of the sequences. So really the best practice is to download the whole thing and either work with that or download the whole thing and then subset it at random using different means that we'll talk about it later on this semester. Let's say we had a project number from a paper that we were interested in. So let's say this paper here looking at the 2014 Ebola outbreak. And we wanted to take now a project number and get all the SRA. So find not just what genes do they assemble and proteins do they assemble and make available like we saw at the very beginning, but what was the raw data associated with that project? And often SRA projects have SRA associated with them. And so we can use eSearch to actually do that as well. So rather than providing a nucleocore or a protein database, we can look for the SRA database using our query. And so let's hit enter and see what we get. So sometimes this can take a little bit. There are 891 SRA associated with this project. So this was a very big project. We can then try and use those to get a list of SRA accession numbers that we can then pull down some subset of. So in order to do that, we want to take this output just like before and we want to pipe it to another command, efetch. Now we're not going to fetch the actual sequences like we did previously because the raw data are way too big for that. We're going to fetch information in the format of information on each individual run, because that is also going to house the SRA accession numbers. And so this, for some reason, can take up to 30 seconds. But I'm going to do this on its own before piping it or redirecting it to an output file, just because I want to show you what it looks like. So this looks like a mess, but this is actually in a spreadsheet format. It's a comma separated values, or CSV format. You can see there's some empty cells here, but we've got lots of things, and here are some of the column headers at the top. There's whether it's paired, all sorts of stuff. So let's redirect that to our info.csv is what we'll call it. And so while it's doing that, I have actually already taken the liberty of pulling that CSV file out previously and opening in Excel just so you could see that it is indeed a table. And so if we look here, the first column is run, and we've got all of our SRA numbers, as well as other things like the date it was released, the library names, how it was sequenced, the size of it, the length, how many sequences there were, bases, lots of great information that we could use if we wanted to. And so this is where Shell starts to be powerful. What do we want out of this table? Well, we really just want everything in the first column. We don't really want anything else as far as what comes to downloading the raw data. So we can use a combination of both printing this file back into the terminal, and instead of looking at it, we can pipe it to another command. And in this case, we're going to use cut. Cut will let you cut out columns from a table format. Even if it doesn't look like it here, this is a table and it's got a pattern. We want that first column. So you could man cut and look at what these different arguments are, but just take my word for it for the sake of the video right now. -F is basically the argument for indicating which column you want to keep. And we want to keep 1, the first column. Now, how are the columns separated or delimited? -D. They are comma separated, so within quotes we've put a comma there. If they were tab delimited, you would put a tab. If they were delimited by an @ sign or something crazy, you would put that there. And then we're going to pipe the output to head, just so we just see if it works with the first several lines, because that's kind of a nice way to shorten things. So let's do this one step at a time. We'll cat info.cv. Okay, it's messy, but it is what we want. Now let's cut -F1-D, and we will go ahead and look at it now. Okay, we got the SRA from that column 1, and let's just say we'll just head it just to get fewer. let's say we want to cut out run using grep. So we can do this same thing, but before we head it, we can use grep. I always think of grep as grabbing, but that's actually not what it stands for. But you can grep, and it's basically like using the find function, but much more powerful. So if you are ever in Word and you're looking for a specific, text, you can, go to the find set function, and then you can type it in and it'll find it for you. That's kind of what grep does, except rather than finding just that piece, it returns the whole line in which that occurs on. So a pattern that we can see here is that all of our accession numbers start with SRR. So if we SRR, we'll only get those back. So indeed, that's what we got back. So now that we've got this cleaned up a little bit, we'll go ahead and redirect this to IDs.text. Okay, and you could even head IDs.text. This is what I do. It's always good just to double check every single step you do. Okay, so there's a couple ways we could go about getting all of these data. Again, we're only looking at 10 right now, but it could be 1,000. So obviously, what we don't want to do is type them all out individually by looking at them here. That would take forever. Maybe not if it was 3 or 4, but why don't we do something a little bit smarter instead of harder? So I like to use a command called xargs, or xarguments. It's basically for inputting things in a list format and treating them each as a variable. And we'll talk a lot more about what variables are, and this is probably the most complex thing you'll deal with in this lab or even this week. But I think it's kind of good to get exposed to these ways of thinking early on. So what we really want to do in plain English is we want to say fastqdump this line, fastqdump this line, fastqdump that line. No matter how many lines there are, just go all the way through it, right? So the easiest way to do that, at least for me right now, is we would print that line to the terminal and then pipe that line, so one line at a time is getting piped over in order, to this function called xarguments. Now what xarguments does is it lets you replace anytime you put this $1 with whatever was on that line. So in this case, let's look at this and predict what it's going to do. Here we're saying, all right, we're going to print this line to the terminal, and then we're going to use this xargs line, and this -n1 is basically saying treat each one line separately. So you can just ignore that and just use this thing as one piece. What it's going to say is, all right, we're going to do this command echo and say this is a number and then our variable from the line. So if this works, what would happen is it would say, this is a number, SRR19 blah, blah, blah. This is a number, SRR19 blah, 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 all the way through, no matter how many there are. So let's go ahead and paste that in and hit enter. Okay? You see that worked just right for us. We could, just if you wanted to see what happens if you don't have this -n and 1, it actually doesn't split them up amongst different lines. So here it's just treating the whole list as a single variable rather than one per line. So this is a number and then it listed all of them. That's not what we want either. Okay? So why would I do this as a number first? I like to use echo to test things, to make sure that it's working the way I want, particularly if I'm about to download terabytes of information. I don't want to spend hours downloading the wrong stuff. So that passes kind of my first examination, goes, okay, this is doing what I think it should do. So now let's replace echo with what we really want to do. So I'm not even going to start a new line. I am just going to go back, leave that variable in there, and I'm going to type in what I actually want it to do. Well, I want it to fastqdump, right? Oops. Fastqdump -x, we'll just keep it short. We'll say I want the first 10,000, and I want them split files, and then that variable number. And we'll hit enter. Now it's going to go through one at a time. This is not all in parallel, meaning it's not going to do all 10 of them simultaneously. It's going to do one, and when one's done, it'll do another. Part of this is to not overwhelm servers. It's kind of a polite thing to do, but also because it would be a more complicated thing to write right now for you guys. So I'll be back when this is done. One thing to keep in mind before I go is if you get some errors that kick back here, sometimes that can happen if you Control C'd out of this at one point and stopped it and tried to start it again, if you have an incomplete backup in that SRA folder, in that NCBI folder within your home directory, that can cause errors. So if you start to run into errors after stopping and starting again, go and delete everything within that folder and then try it again. Okay, I'll be back when this is done. Okay, so that took a while to download, but it's all done. And if we type ls, well, we've got all of these files. And just think, we could do this with 1,000 files if we wanted. You could start it and leave for the day and come back and have them all downloaded next day to work on. So this is a nice, efficient way to do lots of things. What if, and I'm not going to make you download a bunch more stuff, but what if we wanted to be smarter about what we are or not going to download rather than just the first 10? What if there was a specific date that potentially there was a spill in the lab and there might have been contamination? Well, we could actually grep for certain dates within that CSV before we cut out the column that has the SRRs and end up with a list of just sequences from that date. So let's go ahead and try that out. Let's go ahead and just grep for that date. And so we've identified all those dates and gotten those whole lines. And then what we would want to do is actually cut out that first column for the SRR, grep for the SRR to remove the run at the top, and then maybe head just the first 10 lines. So this is a different set of sequences, but they're all from that date. You could do this if there were columns associated with certain types of experimental designs that you might be interested in, or you know, whatever you can come up with. You could use something as simple as what we just did to do lots and lots of different things that are, you know, nice ways to break apart your different data in different ways. Last but not least, I think it's good to kind of just review some of the things we've been doing. It's still early on in this class, and you're probably still feeling very lost and confused, which is totally normal. So try and give yourself a little challenge, and let's do a little challenge related to shell basics. See if you can write a shell script that when you execute it will head all the FASTQ files in your current directory. So the first lab we did, we'll kind of cover how to do that, and I'll show you how I would do it. So first things I would do is I would nano and make a new file and nano it basically at one step. And so let's call it head.sh. .sh is the convention for a shell script. You could put whatever you want at the end though, it won't impact it. And so what's a line that would execute heading of all the FASTQ files in that folder? Well, it would be head star.fastq, right? Maybe you have some that are in different formats as far as how they name the back, and so maybe you have some star.fastq. There aren't in this case, but let's just toss that in there. I'll hit Control X and Y to save it. And so now we have that file in here. Let's ls and see where it is. We've got right there, head.sh. Now remember, to execute, we do the // and then the name of the file. However, we need to make it executable first. So chmod plus X for executable, and the name of it. Now we can // head.sh, press enter, and we just headed it. Notice it says at the bottom, because it does the commands in order, it can't do star.fq for reading because there's no such file. That's because there's nothing in this folder that ends in .fq. So it's like, hey, why'd you give me this command? There's no targets, basically. Okay, so that's how I would do that. The next thing is you could pick one of the FASTQ files that you downloaded during this lab and trim it. So you could use Trim-O-Matic, right? And you could do single end like we did before, where you just need to give it an input and an output and then apply maybe a sliding window. So you could say SRR, let's say 1972919_1, and then we could call it, we'll just call this single end out just for shortness sake, .fq, and we could say sliding window. And again, you might not remember all these exact orders. Go back to your resources. Go look at the lab. When we have practicals, you'll have access to them. You're not expected to memorize these things. I only memorize it because I'm, doing it so much with you guys. And we'll say a sliding window of 10 base pairs with a minimum score, quality score average of 30. And we could go ahead and do that. Okay, so we dropped some there. If you're feeling really comfortable, try and see if you can figure out how to do it with paired end reads. So I'll show you in a second. But if you haven't done it, take a stab at it by pausing and doing it by looking at this manual here. So if we go to Trim-O-Matic, we can see the format for paired in. So we're going to start with PE, and the required things are the input file 1, input file 2, so _1, _2. And then we need output file 1 that for things that maintain a pair. So when you're trimming both paired reads, you only want to end up with files that still have pairs. So if you trim reads from one file but not the other, you need to get rid of its mate, basically. And then we'll throw those extra ones, rather than just deleting them entirely, those unpaired mates go into an unpaired file. So that's why there's going to be 4 output files. For the _1s with a pair, _1s that are now unpaired, _2s with a pair, and _2s with unpaired. And so let's go Trim-O-Matic, see if we can figure this out. We'll go that SRR1, what is it, 972.922 _1, and then we'll do the same one, 72. If you have too many things that have too close, like many numbers in a row that are the same and only vary in the last one, you still kind of have to type a lot. So _2, and then we'll go out1 paired.fq. And again, this is a bad example of output file names, as they're not informative, but I'm just doing this for shortness sake. Out1 unpaired.fq, out2 paired.fq, out2 unpaired.fq, and then we'll just do that same sliding window. Maybe we'll do a window of 5 and a minimum quality of 30 across that. And so something happened. Of course, see, even I make silly mistakes. I didn't put paired end at the front. And there we go. And that worked. And so if through kind of this point in the lab, if you were able to kind of think about, figure out, not from memory, how to do some of these things, other than the doing the paired end reads at the same time, that's a little trickier. You're doing pretty well, and so you should be proud of yourself. All right, thanks.